Welcome to Brain Ponderings, your window into the world of neuroscience research, and with conversations with experts in their particular area, subfield, if you will, of neuroscience and brain research. Today, it's my great pleasure to welcome Michael Kreutz. He's head of the Neuroplasticity Research Group at the Leibniz Institute for Neurobiology in Magdeburg in Germany. He also holds a post at the Center for Molecular Neurobiology in Hamburg, where he heads a group studying dendritic organelles, well, synaptic organelles, so these and nanomachines, if you will, at synaptic terminals, and how their organization and function is important for maintaining the health of synapses and for plasticity, maybe things like learning and memory. And there's a lot of implications for what goes wrong if something goes wrong with these machineries in the synapses. Probably not good. So welcome, Michael.
Thank you for having me, Mark.
I was looking at your background. It's pretty interesting. But before, did you have a long-standing interest in science from when you were a little kid?
Not really in nature science. I really had more interest in philosophy. And at this time, I had an interest in psychology, but I was not looking at it from a nature science perspective. This changed then tremendously when I went to university. So I realized that my brain is better suited for nature science than for other things. And yeah, it was a long way, that I studied linguistics, philosophy, and psychology. And I ended up as a protein biochemist and molecular cellular neuroscientist.
Yeah, I can see how that works. You start with philosophy, which is a little bit abstract, uh, and then you think about, well, my brain is generating these thoughts and so on. So how does that work? And then you think about psychology, and then you say, well, but psychology is still a little bit away from actually what's going on in neural networks. And so it's actually not a bad approach. You're kind of going from a global level to a specific level. So it kind of makes sense in a way. Um, okay, so then you did right. So, but your PhD, I understand, was in behavioral neuroscience.
Correct, yeah.
So that was the psychology aspect, or was that in animals?
That was in animals. It was a transition phase.
I see.
Hmm.
Okay, and that was in Germany too, at Ruhr, and then you came to Boston?
Correct, yeah. Spent some time at MIT, then at Max Planck, and so I moved to neurochemistry, looking at dopaminergic neurotransmission. And then in Göttingen, I started to work on CIF, the CIF receptor. That was stress-related work, but
Yeah.
already pretty molecular. When I came to Magdeburg, yeah, I was a molecular biologist, if you want to.
And when we think of synapses, the major focus is on, how does neurotransmitter release occur, and a lot of nice work done on that. And then what happens postsynaptically when a neurotransmitter binds to its receptor, what happens after that. But there's a lot of, things going on. When I took high school biology, I specifically remember my teacher, Mr. Welty. And so we look at a two-dimensional diagram of a cell, and it's round, right? And you got the nucleus and the mitochondria, and um, and maybe a few other organelles. But I was interested in lysosomes when he started talking about that, and he called them the suicide sacs of the cell. This is in the early 1970s before really much was known about lysosomes. Um, and so, but my point is, when we go through basic high school, often we're looking at a sphere and it's not like a nervous system where you got long axons and dendrites and processes are going on a long distance from the cell body and nucleus. Uh, I had David Rubenstein a couple of years ago to talk kind of some basic stuff of autophagy, but could you just give some background on autophagy, what it is, kind of generally how it works?
Well, autophagy is a degradative pathway which goes back a long time. Most of the work initially started in yeast, as you will know, and the autophagy machinery was deciphered there. There are something like 40 proteins involved, so it's rather complex. In neurons, autophagy has adapted to neuronal function in many aspects. We know that autophagy is involved in degradation of very different proteins, even organelles. And under basal conditions, autophagy is related largely to phagy, that it starts at the ER, which is continuous in neurons, in dendrites and axons, but it is most prominent in axons, much less autophagosomes in dendrites. Now, if you ask which kind of proteins are degraded by autophagosomes and why is this necessary? Well, one reason is definitely that you try to prevent in neurites aggregation of proteins. Because these aggregated proteins cause tremendous trouble in many aspects, and this is the path to neurodegeneration. On the other hand, proteins have a certain half-life, so they get dysfunctional after some time. There are many reasons for that. You have to replace them, and it seems that under certain conditions, it's favorable to choose autophagosomes for their degradation. You know that there's a different pathway, the endolysosomal pathway, that can also be used for degradation of proteins. What we don't understand is actually where is it decided which pathway to choose?
Hmm.
Because in principle, many proteins can be degraded by both pathways.
Yeah. So right, so you can have a protein damaged by free radicals attacking it, or maybe there's some problem in its folding when it's produced. So you mentioned the endoplasmic reticulum, the ER, and generally we think about kind of two kinds of ER just based on whether they have ribosomes and are involved in protein synthesis, the rough ER, and then you have the smooth ER, which doesn't have ribosomes. So you mentioned that autophagy often starts at the ER. So it's the membrane of the ER is being used somehow uh,
Right.
to make this uh, phagophore, phagophore that kind of forms and then engulfs whatever is going to be degraded. Um, so does that occur both the rough and smooth ER membrane?
Well, we know more about the smooth ER, which is prominently present in axons. And
Uh, yeah, yeah.
yeah, and I think in dendrites, this is still unclear because in principle, the machinery for autophagy can use very different membranes for phagophore formation. Yeah? So in dendrites, it might be different. Actually, there's good evidence that dendritic autophagosomes and those in axons are quite distinct. In axons, it's pretty much clear that ATG2, which is the lipid transfer protein that interacts with ATG9, a lipid scramblase, that this is important for lipid transfer from ER membrane to the phagophore.
Uh,
And this machinery is obviously present in axons. And the interesting thing about ATG9 is it's the only transmembrane protein in the autophagy pathway. It traffics from Golgi in very distinct vesicles, from synaptic vesicles to the presynapse. It seems to fuse with the synaptic plasma membrane. It recycles. This is very elegant work from Daniel Colon Ramos. And this, yeah, somehow it's at this stage not really proven, but it points to the possibility that this recycling is in an activity-dependent manner linked to autophagosome formation. Yeah. So these are key players, but there are other key players that regulate autophagosome formation at boutons. But the signaling mechanisms are not really understood. And there's one important problem. It is not really clear how and why activity-dependent autophagy might be different from basal autophagy that I was alluding to, that is to a large degree autophagy. There's very good work from the lab of Volker Hauke on this. And this is something that we are interested in. There's a research unit actually that is funded by the German Research Foundation. The name is Syntophagy, where different groups work on different aspects of membrane trafficking in presynaptic proteostasis. And this is one of the questions that we have.
So if there's this activity, so when a neuron fires, neurotransmitter is released from the presynaptic terminal, and that process stimulates autophagy. So one thing that I I've done a lot of work on neuronal stress responses, and when you exercise your neurons, that's a stress on them, just like your muscle cells. So there's, of course, sodium and calcium influx, but there's calcium's taken up by the mitochondria. You get a burst of free radical production. So maybe what's happening is that there's this stress is making more proteins being oxidatively damaged when the neuron's active. And so this autophagy system is accelerated or enhanced because to get rid of those damaged proteins. Um, you also mentioned that the smooth ER endoplasmic reticulum is in axons and the rough ER is in the dendrites. And of course, there's all this work showing you can get local protein synthesis in dendrites. For a long time, it was thought that doesn't occur in axons. Now, I guess it's a little bit controversial. Um, so, what do you think about this? Okay, what if you block activity in neurons? Um, how does that affect autophagy at synapses? Has that been looked at? Like if you, I guess the crude experiment would be to use tetrodotoxin or something to block sodium channels so that the neurons can't fire.
So autophagy goes down. I mean, this is very clear. So if
Okay.
you eat neurons with TTX, I mean, you mentioned that this might be linked to a stress response, autophagosome formation. Um, I think there's something about it. You also mentioned protein oxidation, and all these are problematic events that are, on the other hand, somehow inevitable if you look at synaptic neurotransmission. So I like this scenario. And I think that it has a lot to do with the high metabolic demand. I mean, presynaptic processes linked to synaptic vesicle release, consume an enormous amount of energy. That's probably one of the reasons why mitochondria are so abundant in boutons. Um, uh, I think it's an interesting scenario, and I consider it likely that these processes are linked and that activity, the dependent autophagy at boutons, is somehow linked to mitochondrial function, energy signaling, sensing, but also the sensing of protein damage. And this is a huge enigma, I think. Nobody knows which sensors are involved and how proteins are sorted for different degradative pathways. And that's kind of surprising given the knowledge that we have on presynaptic function.
Well, when you look at under electron microscope at a synapse, or even, I guess, axons, you don't really see lysosomes, or at least I didn't when I looked at them, but I don't know. So, because these axon terminals are way away from the cell body, so it always seemed inefficient to me to like make this autophagic phagophore way out at synaptic terminal and then send it all the way back to the cell body. So what's going on? Is it still thought that they're sent back to the cell body to fuse with the lysosomes?
There's good evidence for that. I mean, this is initial work, I think, also from Erika Holzbauer. And this scenario still stands, so that there's active retrograde transport via dynein along microtubule. The autophagosome increasingly fuses with lysosomes. There's an increase in lysosomal membrane, there's increased acidification. There's also a fusion with late amnosomes. But I think there's, in the meantime, fairly good evidence that lysosomes, mature lysosomes, are present in axons. They are not incredibly abundant, this is true, but they're clearly present. And I think this is more or less believable now. Um, yeah, so, but coming back to your question, I would guess that the majority of autophagosomes are transported back to the soma and fused there with lysosomes.
Okay, so and that's energy dependent too, right? These motors that move them along the microtubules takes ATP. So anything that interferes with, there's a lot of potential for things to go wrong, right? Then, I mean, you can have impaired retrograde transport of these from the terminal back to the cell body. For example, we worked a lot on things related to Alzheimer's disease, and you have this microtubule-associated protein tau that when it's phosphorylated on certain amino acids, it uh, essentially dissociates from microtubules and kind of destabilizes them. So if there's problems with that, that could affect this autophagy process. And there's certainly a lot of evidence for accumulation of uh, autophagosomes in neurons. Randy Nixon and others have shown that. Okay, so let's get back to so you mentioned that at the presynaptic terminal, this ATG9, autophagy 9, the well, doesn't matter, I guess, mammalian homologue of the yeast protein, that
On here.
it's in it's associated with synaptic vesicles.
No,
Oh
it
no. Okay.
seems
Sorry.
that synaptic vesicles are a distinct pool. This is also a recent paper from the lab of Reinhard Jahn. And from ATG9 vesicles, although they're purified to a certain degree. And it seems that they're also segregated in the synaptic
Ah,
vesicle cluster. And it seems that this is somehow linked to phase separation, and that
yeah.
synapsin, this is a very abundant presynaptic protein, plays a crucial role here. Uh,
Okay. And all right. So can you talk a little bit about some of your specific findings from your lab related to
Mm.
What are your contributions to understanding autophagy at presynaptic terminal?
Well, we came to this by a molecular approach. We were looking for interaction partners for larger proton, a RAP gap. It's called CEPA1L2.
Hmm.
And we realized that it not only has Lear motifs, but that it interacts with LC3. And the lipidated form of LC3 is a marker for macroautophagy. We then realized that this protein associates with autophagocytes. And the interesting thing was that it at the same time binds a very prominent receptor, and it's this TrkB. So it seems to link BDNF TrkB signaling to autophagy. It was really a hard nut to crack, and at the same time, the lab of Volker Hauke had similar observations. And then we realize that what we look at are amphisomes. Amphisomes come into existence when late endosomes fuse with autophagosomes. Now, in non-neuronal cells, these organelles are very short-lived. And this has
Okay,
to,
and Michael, sorry to interrupt, but so this is interesting, right? So, and you worked on this. So, brain-derived neurotrophic factor binds to its high affinity receptor, TrkB, and then there's good evidence that you get endocytosis of the membrane, and then this activated TrkB uh, can keep signaling, if you will, and these vesicles are transported back to the cell body and then maybe affect gene expression in that way. And you worked on that, right?
exactly. And what
Yeah.
we found is that the organelle is not the signaling endosome. This is a textbook cartoon that you have in mind.
Oh,
And no,
is
not
that,
be,
oh, I've got Kandel and Schwartz back here. I don't know if they've updated that yet or not.
no. Yeah, so at least in the cells that we look at, and these are hippocampal pyramidal neurons, we think that following maturation of synaptic contacts, the TrkB that is endocytosed ends up on the outer membrane of autophagocytes. And that this is then still capable of signaling, and this is exactly the point. Now, we talked about the not absence, but the low abundance of lysosomes in axons.
Yeah.
And we also mentioned that there seems to be a segregation between the autophagy pathway and the endolysosomal pathway. And this allows these amphisomes to signal, and that's what we found. So they, on their way to the soma, they stop at boutons and they signal. They increase ERK signaling and they promote synaptic vesicle release. And we found how they stop, and this is the protein that I was alluding to, CEPA1L2. This has a RAP gap domain, so it terminates TrkB signaling under basic conditions. When it stops by a bouton, there's increased PKA activity. Um,
Huh.
the RAP gap activity is blocked. And at the same time, this mechanism results in hopping off from the dynein motor. So the amphisome stops at the bouton. And as long as this signaling is going on, BDNF track B will promote presynaptic function. Well, this sounds pretty much like something that is involved in presynaptic LTP.
Yeah.
And it also sounds like something that might have to do with kind of refurbishing or increasing synaptic efficacy. And well,
And is there increase in physical size of the Bhutan through that
this
mechanism?
this we haven't looked at.
Okay.
So it's something that we could look at, but currently we're more interested to look at the signaling function as
Yeah.
such. Uh, We found that this really goes until the amphisome reaches the soma. This is really intriguing, I think.
Yeah, yeah, it is intriguing. And okay, right. So BDNF, there's BDNF has postsynaptic effects too, or at least there's evidence for that. Um, there's evidence that, right. uh, Well, I guess can BDNF be released from both presynaptic terminal and dendrites?
I think that's also kind of a little bit a matter of debate. I don't want to go to
Okay, all right.
but what I can say is that we don't see these amphisomes in dendrites, but there can be
Okay,
different reasons for that.
okay. And so the amphisomes are the membrane there, the lipids are coming from the plasma membrane, not the ER?
Probably, this is a very good question. Uh, you need two membranes for phagophore formation. And I think, or we think that indeed part of the membrane comes from endosomes. So
Yeah,
the
well, that would make sense. I mean, the track B receptors in the plasma membrane.
mm.
Yeah. All right. So let's see, what else do I have here? Ah, what's the role of calcium? My favorite element.
Well, you need calcium for synaptic vesicle release. You need calcium for a lot of things in synaptic neurotransmission. So of course it's mandatory. You don't get activity-induced autophagosome formation without calcium
Yeah.
because it's driving and fueling a lot of processes that are important.
But what specifically in terms of in stimulating local autophagy, for example, phagophore formation, what is it through the classic calcium, calmodulin, the kinase, and then some target proteins?
That would point to autophagy in dendrites, and there's indeed
Oh,
fairly good evidence that you get autophagy formation after the induction of NMDA receptor-dependent LTD, long-term depression. This is work from the lab of Vasiliki Nicoletopoulou. Vasiliki, if you watch this video, I know that I have mispronounced your name again, but so it is her lab who has contributed significantly to this. So you get autophagy induction there, and the calcium influx somehow is then related to activation of the upstream proteins involved in phagophore formation, like ALK1. I think this is not really understood in detail. And yeah, that's a very interesting observation.
yeah. And then there's, was this your paper or another one? So there's evidence in dendrites, okay. So the typical idea is you have lysosomes, they're inside the cell, and uh, the autophagosome fuse and the contents are released into the lysosomes, and then there's proteases that chew things up and recycle some things like amino acids, lipid components maybe. And but there's also evidence for these so-called exocytic lysosomes that I guess the lysosome itself fuses with the plasma membrane and releases content.
That's correct. Yeah, that's what we have published recently. So there's actually also evidence for secretory autophagy. So it seems that under certain circumstances that are not really understood, these organelles confuse with the plasma membrane and they release their content. In case of the exocytotic lysosomes, it seems to be a subpopulation of lysosomes. So not all lysosomes are competent to fuse with the membrane. And we know a little bit, or we learned a little bit about the molecular players and the function. So You need for fusion and interaction of the lysosome with a scaffold beneath the NMDA receptor.
Ah,
This is MAGAC. It's called SAP102. And this MAGAC directly interacts with NMDA receptors. And here calcium comes into play again, because the influx is necessary to change the conformation of this MAGAC.
huh.
And only in its open conformation it will plausibly bind to these lysosomes. Then the lysosome is docked there. And without going into too much molecular detail, it seems to fuse then with the dendritic plasma membrane. And we can only speculate on that, but it obviously releases catapsins. And these are proteolytic enzymes, and they can have a broader or more narrow spectrum of targets. And the most plausible targets are matrix metalloproteases themselves, they get activated, and components of the extracellular matrix. And if you look at that, I mean, the extracellular matrix is covering also spine synapses, and there is a lot of glue on synapses that is
Yeah.
largely visible. But if you want a spine to grow, so induce structural plasticity, this has to go away. And what we think is that this is a very elegant maneuver to, on one hand, to get rid of garbage in a not very sophisticated manner, but um, and probably there's a need for that. I mean, dendrites can be very thin as well, and you sometimes you just have to get rid of it. We think that it will go away from the brain with the lymphatic system, but it has a second function, and this is to support synaptic plasticity. I find well, I have to admit that I said a lot of things that are speculative, but it makes sense, and I find it very elegant. I mean, neurons are very smart cells. They really know what to do in situations that are very difficult. Yeah.
Uh, so I was thinking about, so that we played around, so we didn't do much on this, but the lipids are not uniformly distributed in the membrane, plasma membrane, and where receptors are concentrated, there tends to be relatively high concentration of cholesterol and some other lipids called lipid rafts.
Right.
So what you're describing, it, is there evidence then that these exocytic lysosomes, the lipid composition of the membrane in those regions is what's, I don't know if it'd be directly, but somehow targeting them there? Or is it, is it something about the lipid composition of the membrane there?
We looked at snares, but this is not yet finished and conclusive. But we haven't looked at lipid rafts. I mean, that's an intriguing possibility. I think, yeah. We know relatively little about where exactly these lysosomes will fuse, whether there is a specialization in the membrane, for instance.
Well, this is a, uh, oh, I don't know if you can see this, what's
Only
going
see
on.
the editor, that's you.
That's strange. My background, oh, there we go.
Ah, yeah, membrane micro. Yeah, yeah, I see this. Yeah,
This from a long time ago when
yeah,
I was interested in this. We ended up not doing a heck of a lot on it, but I did this edited volume with some other, that was like, geez, I don't know, 20 years ago or something.
yeah.
That's an interesting thing. Let's see. What else was I going to ask about? Right. So again, I think it's the same idea though that when when glutamate binds to its receptor, you get calcium influx, free radical production. So there's a lot of stress going on at dendrites when they're activated by glutamate, and maybe this has to do with a very efficient mechanism to prevent accumulation of uh, damaged proteins or
I think
aggregate.
that it's not necessarily damaged proteins that go out there,
Ah,
uh, because we linked it largely to chaperone-mediated autophagy. These are
ah,
proteins that are, yeah, if they reach these are high bond proteins. If they reach a critical concentration, it's a problem because they start to aggregate proteins like tau, huntingtin, and others.
yeah.
And so they are still soluble at the time when they are incorporated into lysosomes. So this might be a preventive mechanism
Yeah.
before the damage was done.
Tau's interesting, right? Because it's normally in the axons and it's absent or sparse from the dendrites, and but in Alzheimer's disease or frontotemporal dementia, it accumulates in the cell body and dendrites, and maybe that has to do with microtubule depolymerization or something in the axons. But so the point there is that at least with tau, it's not usually present in the dendrites, and maybe so there's some the dendrites are recognizing tau as something that shouldn't be there, and therefore spitting it out, getting rid of it.
I mean, this seems to correlate with the phosphorylation status of tau. We haven't looked at that. But tau also shows up in spines. I mean, it's present there even under physiological conditions, but it causes early synaptic dysfunction in
Yeah.
Alzheimer's disease. And yeah, this is definitely one of the yeah, top proteins in disease progression in Alzheimer's. And yeah, it seems to be linked to amyloid beta signaling, what tau is doing. So in the last 10, 15 years, I think a picture is emerging that shows the interaction of both. But yeah, I don't see currently whether CMA, chaperone-mediated autophagy, is really protective in Alzheimer's disease. I think that's just, I don't know. But there are people that know much more about it and have done tremendous work. You mentioned Ralph Nixon, also David Rubenstein and others. So,
Now, when I think of chaperones, protein chaperones, I think of heat shock proteins, for example, or in the endoplasmic reticulum, there's one we studied somewhat called GRP78, glucose regulated protein 78, and its role prevent, it used to be called BIP, prevent protein misfolding and we mentioned that the endoplasmic reticulum in the dendrites uh, that can have ribosomes and synthesize proteins. Um, I guess what I'm asking is if one were to manipulate, say, genetically or physiologically levels of, say, GRP78, and there's actually an easy way to do that, even in cultured neurons, you just add 2-deoxyglucose, and that's how it was discovered. It'll cause a huge increase. So you're impairing energy metabolism, and then GRP78 goes up, and we found it's neuroprotective. Um, so I don't know. It'd be interesting if I were I wasn't retired, and maybe after talking to you, I'd have someone in the lab, or maybe we'd collaborate, right? And test the hypothesis that GRP78 is modulating dendritic, exocytic, um, well, yeah, exocytosis of these bad accumulating protein.
That sounds interesting. I think there will be sensors, and it's very likely regulated, but we don't know much about mechanisms.
Yeah. And you
And
mentioned energy metabolism, so that might be an interesting one.
this one, or ER stress response, or there are many factors that could influence that. And yeah, I mean, there, there's fairly good evidence that there's a distinction between spine and shaft synapses. And NMDA receptors are also found in shaft synapses. And this could be differentially regulated in disease conditions as well, because the
Huh.
spines are pretty much sealed. They're a biochemical microcompartment, whereas the shaft synapses are naked. They have to stay in the dendrite and they're exposed to all kinds of things.
Interesting. Yeah, that's an interesting idea. Well, you can, since a lot of American scientists are leaving the United States now, and actually this is it's true because of the dramatic reduction in funding by the NIH and
Mm.
NSF. Maybe you can get an American postdoc to do some of these experiments.
Sounds good. Yeah, it's very serious. It's not funny at all. What's
No,
going
it's not funny, but yeah, I don't know. But my son just got his PhD in biochemistry, and so he actually had a job lined up at the FDA, and that's on hold now. Whoops. Uh, okay.
on? You're a bit frozen currently.
Yeah, let's see. There, is it okay now? Is it okay?
You're still frozen. I can hear you.
Okay. I don't know
What's
why I'm
better?
frozen. Maybe it'll come back. All right, that's okay as long as we have the sound and we can see you moving. It's okay. I was going to ask you about um, the phenotypes, the neurological phenotypes of mice in which genes for select, I guess, autophagy-related proteins are disabled or knocked out. So, for example, I guess two of them we haven't mentioned much are ATG7 and ATG5. So what happens if those are knocked out either in the whole mouse or if these experiments have been done in, say, only neurons or certain neurons?
Well, if you knock out autophagy genes, the phenotype is usually during development lethal.
Yeah.
And in the brain, you find massive neurodegeneration. And if you do this in a cell type specific manner and at later stages of development, the phenotype is more complex. For ATG7, it's for instance shown that it has a tremendous impact in, I think, Purkinje cells in the cerebellum, but not so much in other neurons, which is kind of weird. With AGG5, what you see is increased excitability. There's
Hmm.
ER release of calcium. It's a ryanodine receptor mediated phenotype
Huh.
that is particularly prominent at certain synapses. And so there is evidence that mutations in autophagy genes causes disease in humans. But it's this is my view. I mean, perhaps it's naive, but I'm surprised that there are relatively few cases reported. It might be that the deletion or loss of function of autophagy genes is just not compatible with life.
Yeah. All right. So critical. Is there any way to selectively disable autophagy and axons and not dendrites, or to locally, how would you do that? I don't know. That'd be,
I'm thinking
yeah.
selectively. Well, there are obviously ways to interfere with protein function. So you can use techniques to damage protons. But this all comes with overexpression or knock-in. So it's hard to target specifically an endogenous protein, but that's a way to go. And we know a little bit, not much, on the regulation of autophagy at pre-synapses. So, for instance, this is a relatively prominent example. It's work from Craig Garner and Eckert Gundelfinger, who were involved also in the identification of major protons of the pre-synapse. And two of them, these are giant protons, these are piccolo and boson.
Hmm.
And boson seems to directly interact with ATG5. And what is interesting is that it inhibits ATG5 function. We don't know exactly how. It might just sequester the protein or inhibit it by other means.
Hmm.
And we know the domain that binds. So it's a certain domain in boson, and you could use this to try to interfere with an interaction that will selectively happen at boutons. That would be another strategy. But apart from that, I don't know really the answer. I would be grateful if you come up with a suggestion. That would
Well, I
be.
just thought of something actually, but uh, remember the squid giant synapse?
Mhm.
Very important in the early days of uh, uh, neural biophysics, I guess you'd say, electrophysiology. And what about take that and where you can inject antibodies to specific autophagy proteins? Because you can actually inject, right? Physically inject into a squid giant synapse and then what? But that's going to take a lot of, I think, background work to.
Well, we would have to establish in our facilities
Yeah, yeah.
that
I,
we
I
can.
I don't
It's
think many people are working on with that anymore. 'Cause I don't. Yeah.
right. Yeah.
Okay, last thing. Uh, Golgi satellites and LTP had a nice paper in Cell Reports a couple years ago. Uh,
Mhm.
Can you talk a little bit about that?
Yeah. So we discovered some years ago, I think almost 10 years ago, that Golgi is not only present in the soma, and that there are satellites that are different from Golgi outposts. These outposts were described previously, but they are mainly at branch points in dendrites, and they play a role in development. Whereas the Golgi satellites that we found are present all over the place. So they are in all dendrites, apical, basal dendrites, third order. So and these are interesting structures because they are capable of glycosylating membrane proteins. And you could see that synaptic transmembrane proteins pass through Golgi satellites. And what was extremely interesting for us, these are very small vesicles. They are not huge, so they have limited capacity. We found that they are mainly involved in cellulation, which is a kind of post-translation modification that is specific for relatively few proteins.
Hmm.
This is most prominently NCAM. Uh, and we could show that this indeed has, there's a link. And the other form is fucosylation. Now, fucosylation has been studied a lot in the 70s and 80s, probably because there were probes and lectins available to look at them, but not so much in the past 20 years. But we found that the entire machinery is there in Golgi satellites. Now, fucosylation of synaptic transmembrane proteins has been linked to LTP induction. And well, if you think about it, most membrane proteins in dendrites don't show mature glycosylation. And what we think is that following a certain type of stimulation at synapses, these proteins can be endocytosed and they traffic to Golgi satellites. We also showed how this works, so retromer is involved. And they can potentially even go back to the ER, but through their passage through the Golgi satellites, they get glycosylated locally. So it's a very local thing. And this definitely will impact on function, but also on protein half-life. So proteins that are glycosylated can stay for much longer, for instance.
Huh.
And yeah, so we know that Golgi satellites are also present in axons. And this brings you then to a number of novel questions. If you go back to autophagy, ATG9 is going through Golgi. These vesicles, I don't know of any data that show that these ATG9 vesicles can fuse with Golgi satellites, but that would be also a very interesting scenario. And um, it points to something that I think is very important for neuron cell biology, and this is neurons have had to solve the problem that they had to serve the needs of something like 10, 15,000 synapses almost at the same time. And the logistics for that are kind of the demands, this is crazy. I mean, if you look at the length of axons and the complexity of the dendritic tree, to organize all this from the soma also doesn't make any sense.
Right.
So they have their logistics for what they immediately need in place. And the main aspect is everything has to serve synaptic neurotransmission. And in humans, this has to work for 90 years, 100 years. So I think that this efficient logistics of protein trafficking, replacement, and degradation is the reason why neurons can survive for such a long time.
And so I was thinking, so you mentioned NCAM, a cell adhesion molecule, and Gerald Edelman's hypothesis on memory. Do you remember that? Where his idea was that that memory is encoded by, I think this was the idea that by increasing the stickiness of one neuron to another, or so the extracellular matrix or the cell adhesion interactions were important for learning and memory. And so this finding of yours suggests a potential molecular mechanism that's consistent with that, or am I looking at this wrong?
Well, this definitely has impact on the surface expression and the interactions. We had in this paper that you were alluding to experiments where we used knockout mice. These mice were devoid of Golgi satellites, at least they were significantly reduced, and they had a strong LTP phenotype. What was interesting, the phenotype was strong in the most distal dendrites,
Huh.
where, if you think about it, this is several hundred micrometer away from the soma. And it could be that the function of Golgi satellites for encapsulation is indeed something that keeps these synapses in good shape and going. And whether this is directly part of engram formation, I don't know. I mean, this is far-fetched.
Okay. It's far-fetched right now anyway. Um, right. So, I remember some illustrations of kind of the organization of synapses, say on a hippocampal pyramidal neuron, dendrites, right? And where the glutamatergic inputs were the most distal, and then more proximal, maybe cholinergic, or serotonergic, and then, close to the cell body, the GABAergic inhibitory. So do you think this is only these Golgi satellites are specifically playing a role at glutamatergic synapses and not so much at other synapses?
We haven't looked at that. So this is something that we simply don't know. Uh,
Okay.
um, yeah. Well, inhibitory synapses in dendrites are usually very close to spine synapses.
Yeah.
And
Oh,
I guess they could serve both.
yeah, yeah. Ah, right. Okay. Let's see. And what's the distribution of NCAM if you look distal to proximal on dendrites? Is that known?
Oh, I
Don't know. Oh,
probably yes, but I don't know the answer.
okay. I'm just wondering if there's a relation to that. And did you look at learning and memory in this paper where you saw a, a negative impact on LTP of
No, this was no, we didn't. I'm just trying to recall, but there's no behavioral experiment in this
okay.
paper.
Okay.
No.
So that's something I guess to do.
Mhm.
Um, right. And then I said last before, I've been interested in we talked about metabolism, and one thing I've been interested in is mTOR pathway, right, which is kind of negatively correlated with autophagy in general. In other words, you inhibit mTOR with rapamycin, that tends to increase autophagy. Um, have you done like crude experiments like that to see how rapamycin affects some of these processes you're looking at in presynaptic terminals and dendrites?
That's a very good and interesting question because we and many others we had quite some difficulties to induce autophagy with maneuvers like starvation, rapamycin in neurons.
Ah,
And there are very mixed results. And we then decided as a group, I mentioned the research unit, Syntophagy. So there are different groups, different labs. And we decided to do the same experiment in every lab, to use the same batch of rapamycin. And it was incredibly variable. So what I'm saying is that something is unique about the induction of autophagy in neurons. Same with starvation. And
interesting.
sure, what to do with this. I mean, there are obviously differences in culture conditions and so on, but I think it's important to reach a consensus, an agreement on which are good experiments to induce autophagy in neurons, and that's the reason why we did this. But I'm not sure whether rapamycin-induced autophagy will indeed reflect at least activity-induced autophagy.
Yeah.
I think that this is the case. And one has to be a little bit cautious because the autophagosome content profiling has shown that there's probably more than one type of autophagosome and that the content can be very different depending on many factors. And whether rapamycin is really inducing the pars pro toto autophagosome for a neuron, I would actually doubt this.
Ah, interesting. That's really interesting. So definitely regulated by activity in the neurons, not so clear on the relationship with the mTOR pathway in neurons. Or it could, in theory, it could be,
I don't doubt that there's a relationship with the mTOR pathway.
yeah,
It's
okay.
just that the maneuvers that we can use to induce autophagy or yeah, it's difficult.
Amino acid deprivation
That some people report positive results, others
okay
not. That's,
2-deoxyglucose
I think that that was only one paper, and yeah, positive.
okay all right but anyway the take-home message is keep your neural networks active.
Sure.
Yeah,
And the neuron will take care that your synapses are kept tidy and clean.
that's right. Uh, so during this podcast, you and I have been cleansing our synapses, and hopefully the viewers and listeners will focus on what we're talking about, challenge their neurons a little bit,
Right.
clean them up. Okay, thanks a lot for taking the time for this, Michael. I enjoyed meeting you and talking with you. A fascinating area of research.
Yes, thank you again for the kind invitation. I also enjoyed talking to you. And yeah, stay tuned.
Okay, I will. Yeah, that's why I'm trying to keep my brain active by doing these podcasts so I can read up on what people are doing in areas that I'm not focused on so much. It's a lot of fun and hopefully this is useful for the viewers and listeners.
Okay.
Okay, bye.
Bye bye.